All right, our next speaker is Joy Yang from Massachusetts Institute of Technology, and she's a student in computational and systems biology under Martin Polz. Did her practicum at Lawrence Berkeley National Laboratory in 2016, and she'll be speaking about statistically identifying the mechanisms of phage-host interactions in the Nahant Collection. Joy.
Cool. Thanks for having me here and for having fun in me for 4 years, first of all. So basically, we're talking about the Nahant Collection, which is a beach that's in really close to MIT, and so we can literally drive to the beach and collect samples. But first of all, since this is a really broad audience, what are phage and what are their hosts? So bacteriophage are viruses that infect bacteria, and why do we care about them? Why do we care about them in seawater? So in 1 milliliter of seawater, that's about like a thimbleful, we have like 1 million bacteria and 10 million phage. And so considering that there are 36 million kilometers cubed of water in the top 100 meters of the sea, that's a lot of bacteria and a lot of phage. And so rare events for a single cell does not equate to rare events within a population. So these bacteria and their phage are constantly meeting each other, and they're constantly evolved in this arms race that they have against each other. And so a lot of things need to happen in order for a successful infection to occur. So first of all, there needs to be successful binding of the phage to the receptors, and then once they inject their DNA, this needs to undergo successful replication, transcription, and translation. And at various points, it can be cut up and digested. And a lot of these processes and a lot of these enzymes that have been discovered through phage research are now used in biotechnology. And so this is kind of like a fruitful area of research to go into, even though it's very, very much basic science. So in the Polz Lab, we work with the Nahant Collection, and so this is the largest phylogenetically resolved phage-host cross-test. And what I mean by that is Kathryn Kauffman in our lab collected 250 bacteria from the Nahant beach, and she also collected 250 viruses, or 243 and 241 actually, but around 250. And she tested all viruses against all bacteria, so we know exactly which viruses can infect which bacteria. And we also have sequenced all the genomes of the bacteria as well as all the genomes for the viruses. So we can reduce this data down to 1,000 phage gene clusters and 10,000 host gene clusters and try to statistically infer what genes might be responsible for infection. And so my thesis has been really kind of like aim 1 is just exploratory data analysis, what can we find? And aim 2 is more zoning in on a specific interaction. And aim 3 is a little bit of the communication aspect that we talked about a little bit this morning of developing curricula for statistics outreach. So I'll mainly cover aims 1 and 3 today. So aim 1 is identifying novel mechanisms of infection and defense in the coastal oceans. So basically this is, okay, now we have all this data, we have all the sequencing data, what do we do with it? Because we can't just look at it, so what do we do? So some really simple techniques that we can use are just like simple regressions to try to figure out what genes might be responsible for infection. And so, for example, this is one phage that infects all these bacteria. We can try to regress all the bacterial genes against this phage infection profile to see what genes might matter. And so the problem we get when we're doing this, though, is that we have a lot of structure in our data, and so there's a lot of phylogenetic confounding. What that means is, so for example, this is like a bloom, so all these bacteria are really closely related. This is like if you do a heart disease study and then you recruit 18 siblings, you don't want the genes from these siblings to really outweigh all the rest of the genes because these are population confounded. And so basically all the genes that are present in this population pop up as negative effects just because we have so many of these bacteria, but in reality it's just because it's population confounding. And there's actually a really neat trick that you can play. So in statistics it's just called generalized least squares, and in signal processing it's called whitening. So you just multiply by the inverse square root of the covariance matrix, and then you end up whitening out your noise. And so now, if we do this, instead what we get as negative effects are these genes that are in closely related hosts to this group of hosts, but can't be infected by this phage. And so that's like if you had twins in your study and one twin had a heart disease and the other twin didn't, then you're more confident that these differences might be responsible for heart disease, as opposed to if you had two completely unrelated individuals. So that's kind of encouraging, and it's kind of showing that maybe this is like the right way to go. So another thing that you can do, like a slightly more complicated model, is just using a multivariate model. And so what that means is we're going to throw all of the virus genes, all the host genes, and possible virus-host interaction terms into our model and try to see if that ends up fitting our data. And the problem with this, though, is that we have 1,000 virus proteins and 10,000 host proteins. And so that means that we have 10 million putative interaction terms. And since our matrix is 243 by 241, we end up with like 58,000 observations. And so that's a 58,000 by 10 million matrix, which is a lot. The problem is, even if we could compute on this, how are we going to interpret 10 million coefficients? That's like pretty uninterpretable. So how do we tackle this? It turns out that what we ended up doing is kind of doing a pre-filtering step using generalized least squares, and then we have like a lot less host and phage terms and putative interaction terms. And then we can plug this into our model, and then I didn't copy and paste two images twice. This is actually the predictions versus the real data. And that's great. We can massively overfit our data. But as they say, all models are wrong, some are useful. Is this model useful? Can we make any sense out of it? And even this model with like a smaller number, it's like 60 phage terms and 112 host terms, is a complete mess. It's like a complete hairball. So how do we make any sense of this? And one idea is when we were trying to fit the model in the first place, one idea that my committee member had was, well, what if you use alternating minimization? So essentially what the model boils down to is this virus matrix times this interaction term matrix times the host matrix, and what we're trying to fit are the interaction coefficients. And so what if we decompose that into two components and solve for each individually? That reduces the size of our problem, and maybe that makes it feasible. But we didn't end up using that, but the interpretation is kind of nice, because now if we have orthogonal components for this coefficient matrix, then maybe we can try to use these orthogonal components to make sense of our data, and so or to make sense of our model. So if you take orthogonal components from A and B, the first component ends up being the intercept, just because most infections aren't successful, so that sets the intercept really low. And then if you take successive components, you end up picking out this big infection module right here, and what seems to be the signature, which is the DNA polymerase. It's just a really efficient polymerase, and then we end up getting a lot of proteins that are indicative of what it can infect. So those might be putative receptors, and then you keep on picking out successive modules, and most of these seem to be receptor or maybe tail fiber interactions. Okay, so this is kind of nice, but at the same time, it's not entirely exactly what we want. And what we really want is a good way for my lab mates to explore the data and to actually generate testable hypotheses. And this is kind of a silly slide, but I just wanted to include it because I thought it was so amazing. So this is from John Tukey's exploratory data analysis. And as you can see, these are ASCII density plots and actual hand-drawn density plots, but this is like really what was state of the art back then, right? Like tracing paper. But now we actually have like much cooler toys to play with, and so I just wanted to do like a kind of like a little demo of something that I've been trying to work on to do exploratory data analysis. And so basically, one question that comes up a lot is, okay, let's say that I'm interested in one particular phage. So if I'm interested in this phage, what bacterial genes are responsible for allowing the infections to occur, and what are inhibiting these infections? And so if we select one phage, for example, maybe this one, this is a phage that one of my lab mates is really interested in. And if you zoom in on the proteins that pop up as highly correlated with infection, you end up getting a lot of type 1 restriction modification proteins. And what those are phage defense islands. And so basically these are the proteins that cut up the phage DNA once it's injected. And so if you select some genome that can't be infected by this phage, you can zoom in on the negative effects. And so, for example, here's a really high island. So these are, the y-axis is the negative log of the p-values. And so basically, for this one, you see all these type 1 restriction modification islands. And you also see these like putative integrase proteins. And so this looks like a phage that integrated into the host genome at some point and now is defending the genome against invasion by other phage. And you can zoom in on other islands too, and these all actually tend to look like putative integrases and type 1 restriction modification systems. And so these really do look like defense islands, and that's kind of a story that we're trying to develop a little bit more right now. But if you select other phage, you end up getting things that are more along the lines of positive effects, and if you zoom in on those, they tend to be putative receptors, were enzymes that add modifications to the proteins that are receptors. And so in this case, you get like a pyruvyl transferase enzyme, which is like kind of encouraging that maybe these models are doing the right things. Okay. So now, I guess just as a brief summary of the section so far, correcting for phylogenetic confounding allows us to pick out mechanisms that would otherwise be lost among spurious correlations. The multivariate model allows us to view the problem from a prediction perspective, and it also helps us think about putative protein interactions between phage and host. And visualizing the data allows us to generate more hypotheses about putative receptors and defense mechanisms. So that's the first section, and then I just really want to briefly talk about statistical education, just because this is something that I feel really strongly about. I've been involved in a few outreach projects over the course of my PhD, and one that I'm really lucky to have been involved in is this program called SEED at MIT. And so basically what this is it's a program that's targeted towards underprivileged middle schools and high schools within the Boston area. And they're brought in with a 5.5-year commitment. So they technically are supposed to stay for 5.5 years, but of course there's people who trickle in and trickle out. But essentially what this is they get an hour of academic mentoring in the morning, and then they get 3.5 hours of a project-based class. And what I was responsible for teaching was environmental engineering. And this is because my advisor is in environmental engineering. And so basically, there's a lot of really cool things that you can do to integrate statistics into environmental engineering. So for example, Jesse Krell's lab in our building, they work on air quality monitoring, and so they helped us build these like little, really simple air quality sensors that the students were able to walk around with. And then reading graphs is a huge challenge for 8th graders, but like since these are data that they actually collected themselves, they can actually walk through the data and be like, oh, this is like when we were walking by the street and all the cars were passing, and this is when we were in that taqueria, and then there was all that smoke, and so they can like effectively start telling the story about their data. And we can also teach like experimental design. So I know like George Box taught experimental design to engineers using paper helicopters. So since we're in environmental engineering, we're using wind turbines. So it's kind of fun. They can build their own wind turbine and see what affects it, like whether it's like the blade length or maybe the number of blades or something like that. And we can also do like interpreting maps, because that's also a challenge. And there's a lot of maps of environmental data, and we can do population modeling using game theory. Turns out Prisoner's Dilemma is actually really intuitive to 8th graders, and rock, paper, scissors, that's a game that 8th graders definitely know how to play. And so yeah, I have to really thank Anjali Jain, who's one of my building mates, for introducing me to this program. And so finally, I need to thank my advisors, Martin Polz and Libra Kelly, for having been really supportive throughout my PhD, and also for my amazing lab mates, especially Kathryn Kauffman for collecting the huge, amazing data set, Fatima Hussein for a lot of experimental support, and MIT Parsons, which is the environmental engineering department at MIT, and especially Anjali for introducing me to the outreach program, and pretty much really all of Parsons for having been like great social support, and my committee and my practicum lab for having taught me how to do experiments.